Hello everybody. Today our topic is Mendel's laws of inheritance. In the previous lectures we discussed the law of paired factors and the law of dominance. Now this lecture will focus on the law of segregation. Let's move to law of segregation. The law of segregation or also the law of separation, it's also known as law of purity. So this law of segregation, it states that every individual organism contains two alleles for each trait and that these alleles segregate or separate during meiosis such that each gamete contains only one of the alleles, or in other words we can say that law of segregation states that the two alleles of a gene separate during meiosis or separate during gametogenesis. I will make you understand. If we have a diploid cell, and we have a gene T that's responsible for the height of a plant, so it controls the this T, it controls the trait height. Height is a trait, so this height, it is controlled by gene T, and this gene T has two alleles. One allele actually is from one parent and another allele is from another parent. Now we have two alleles for a particular trait. This is what actually the law of paired characters, the law of paired factors states, that each gene has two alleles. One allele is from the maternal side and one is from the paternal side. So what the law of segregation says, that these two alleles during meiosis, during gamete formation, these two alleles, they randomly move to two different gametes. They move to two different gametes, two different haploid cells. Now this they will move to two different gametes. For example, this capital T in red color, it will move to one gamete and this will move to another gamete. This is basically what law of segregation states. Now let's try to understand this law of segregation with the help of a monohybrid cross. Now, what is a monohybrid cross? A monohybrid cross is a cross, it is a cross between two plants. It is a cross between two plants of same species. Plants of same species. It is a cross between the two plants of same species, considering the transmission of only one trait, considering the transmission of only one character. Now we can write it down. Monohybrid cross is a cross between two plants or two individuals of same species, considering the transmission of only one character. Now for example, a cross between a tall pea plant and a dwarf pea plant, it is a monohybrid cross because only one character is under consideration, and that's the height of the plant. One plant is tall and another is short or dwarf, so the trait under consideration is only one, and that is height of the plant. So in monohybrid consideration we have a cross between two individuals and only one current character is under consideration, and transmission of only one character is under consideration. Now let's try to understand the law of segregation with the help of this monohybrid cross. Now we have two plants. We have a tall pea plant and we have a dwarf pea plant. Both of these plants, they are homozygous for the trait height. The tall plant has the genotype capital T, and the dwarf plant has the genotype small t and small t. Now, if we see this tall plant, it has genotype capital T and capital T. Both the alleles are capital T. Both the alleles are similar. So it is homozygous. It is homozygous. This plant is homozygous. Homozygous. And the alleles are dominant alleles. So it's homozygous dominant. Homozygous dominant. Similarly, in case of the dwarf plant, we have small t and small t. It's again homozygous. It's again homozygous. And we have small t and small t, and these alleles, they are recessive. So it's homozygous recessive. So here we are crossing two plants. One plant is tall and another plant is dwarf. The tall plant has the genotype capital T, and the dwarf plant has the genotype small t, small t. Okay, now what law of segregation states? Law of segregation states that the two alleles that are responsible for a particular character, they separate during meiosis. Now these two alleles, capital T and capital T, they are responsible for the height of this plant. That's why this plant is tall. Now what law of segregation says, that these two alleles, this capital T, they will separate during meiosis. As the gametes are formed, these capital T and capital T, they will move to different gametes. Very important. This is what law of segregation states. Again, in case of the dwarf plant, the two alleles, small t and small t, which control the dwarfness of the plant, they will move to different gametes. Small t and small t. This is what law of segregation says, that the two alleles of a particular gene which are responsible for a particular character, which are responsible for a particular trait, they control the transmission. They control the transmission of the particular trait. They separate during meiosis. Now these capital T and capital T, they have separated during meiosis. They have gone to two different gametes. These small t and small t, they are into different gametes. Now now we are crossing these plants. If we fertilize these gametes with these gametes, the gametes of the tall plant with the gametes of the dwarf plant. So let's make a chart here. We'll cross them. Let's make a chart here. So here we'll put this capital T and capital T on one side, and small t and small t on this side. Okay, if we cross them, we will have this capital T and small t here. We will have capital T and small t here. We'll have capital T and small t here. We'll have capital T and small t here. Okay, now all the plants have the genotype capital T and small t, and since capital T, it is dominant toward the small t, the phenotype will be that of the capital T, and that's the plants will be tall. All the plants will be tall. Now we had two parents. One parent was tall and another parent or another plant it was dwarf. Now when we cross them, all the plants in the F1 generation are tall because they have the genotype capital T small t capital T small t, and since capital T is responsible for the tallness of the plant and is dominant toward the recessive allele small t, all the plants will be tall. Now what is important here? Here in case of the parents capital T and capital T, they were together, small t and small t, they were together, and as per the law of segregation, capital T and capital T they separated, and small t they also separated, and capital T and small t they join it together to form a new plant, a heterozygous plant which has one capital T and one small t, and the plant is tall. This is heterozygous plant. Now law of segregation is proved here. This F1 heterozygote, the development of this type of a heterozygous F1 plant confirms the law of segregation, because this capital T, it has separated from this capital T, and this small t, it has separated from this small t, and they have joined it together to form an offspring which is capital T and small t. So law of segregation says that the alleles that are responsible for a particular character in a plant, they separate during meiosis. This is what law of segregation proves. Now if we go further, we take the F1 heterozygous plants which have the genotype capital T small t. Here we have two plants. Now they have the genotype capital T and small t. Another plant here capital T and small t. We have these two plants, will subject the F1 plants to selfing. Now when we cross them, what we observe in the F2 generation, that we have three plants that are tall, and one plant we have dwarf. Let's try to understand again. We took the F1 plants and subjected them to selfing. Both the parents were having the genotype capital T small t, capital T small t. We crossed them. What we got in the F2 generation is that we got three tall plants and one dwarf plant. Let's try to understand what happened during this cross. Actually, during meiosis, these two capital T and small t, during meiosis or during gamete formation, they separated and they again moved to two different gametes. Capital T and small t, they moved to two different gametes, and this capital T and small t, again they move to two different gametes. Now if we cross the gametes, we will have three tall plants and one dwarf plant like this. We'll make a chart here. We'll make a chart like this. Now we'll put capital T on one side, capital T and small t on one side, and capital T and small t on the other side. We cross them. We have here we'll have capital T and capital T. Here we'll have capital T and small t. Here we'll have capital T and small t, and here we'll have small t and small t. Now if we analyze them, this plant, it has capital T and capital T. Both alleles are dominant and this plant will be tall. Okay, now we have two plants which have genotype capital T and small t. These two, they have capital T and small t. Since they have one dominant allele, these two plants will also be tall. And we have one plant which has the genotype small t. This plant, it will be dwarf. Now both the plants were tall, but in F2 generation we have one plant which is dwarf. So what was in these two, what was in the two tall parents that led to the production of a dwarf plant in the F2 generation. This is explained by the law of segregation. Here, actually, the two tall plants, the two tall parents, they had this allele, small t. And this small t, it was present together with the capital T. So its effects, its phenotype, it was suppressed by this capital T. And when the capital T and small t, they separated as per the law of segregation, they moved to two different gametes. And then the small t, it combined with this small t to produce this offspring, small t and small t. So this is the how the law of segregation works. The two alleles that are responsible for a particular trait, they separate during gametogenesis. Now let's quickly look at the genotypic ratio and the phenotypic ratio of for the monohybrid cross. Okay, let's see what is the genotypic and the phenotypic ratio for the monohybrid cross. Now see here, we have three tall plants, this capital T and capital T, this one. We have another two tall plants, they have a genotype capital T, small t, capital T, small t, and we have one dwarf plant that's small t and small t. Here you can also check it here in this box. Now, how many plants are tall? If we look at the phenotypic ratio, if we look at the phenotypic ratio, how many plants are tall? So tall plants are, we have three tall plants, 1, 2 and 3 are in this box. We can see here TT will be tall, this TT and this T, capital T and small t, they will be tall. So tall will be 3. How many plants will be dwarf? How many plants will be short? Only one plant is dwarf. So if we look at the phenotypic ratio, the phenotypic ratio is based on the external appearance of the plant. So we have three tall plants and one short or one dwarf plant. So phenotypic ratio is 3:1. Now if we look at the genotypic ratio, we look at the genotypic ratio, we look at the composition of the alleles, composition of the genes, we look genotypic ratio. Here one capital T and capital T, how many times we have capital T and capital T? We have only once, capital T and capital T. How many times we have capital T and small t? We have it twice. And how many times we have small t and small t? Small t and small t, it's again once. So the genotypic ratio will be different from the phenotypic ratio. While as the phenotypic ratio is 3:1, the genotypic ratio will be 1:2:1. So the genotypic ratio and the phenotypic ratio of a monohybrid cross, they differ, while as the phenotypic ratio is 3:1 and the genotypic is 1:2:1. Now we'll take another example to understand the monohybrid cross. Now this is another cross. It is the cross between two pea plants. One pea plant, it has green color pods, and another pea plant, it has yellow color pods. Now this is one plant and this is another plant. This plant, it has green color pods and the genotype is capital G, and this plant, second plant, it has yellow color pods and the genotype is small g, and phenotype it is yellow. Now this capital G or small g, they control the trait color of the pod. So the character or the trait here is the color of the pod. So capital G is responsible for the green color pod and small g is responsible for the yellow color pod. And both of these two plants are true lines and they are homozygous. Here we have both alleles are similar, capital G and capital G. In case of the yellow color pods, we have small g and small g. Both alleles are similar, so it's again homozygous. And what law of segregation states that during meiosis, these two alleles, capital G and capital G, they will go into two different gametes. They will go into two different gametes. Similarly, in case of the yellow color pods, the small g, they will go into two different gametes. And as in case of the previous slide, which in case of the cross between the pea plants that we saw in the previous plants, and the correct trait in previous slide was the height of the plant. Here we will again have all the plants will be green color because capital G will be dominant over the small g. Again, if we cross them, you can cross them like this. You have capital G and capital G on one side, you have small g and small g on the other side, and you cross them, we'll have capital G, small g, 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 and capital G, small g. So all plants will have the phenotype capital G, small g, and all the F1 plants, they will have green color pods. Now here I need to add one point, that this chart which we formed, this chart, it is known as Punnett square. This chart is known as Punnett square, Punnett, Punnett square, and it is named after a scientist, Reginald Crondell Punnett was a scientist. Punnett was a scientist, and because of his name, this chart is also known as Punnett square. He gave this chart actually, and this Punnett chart, it is used to predict the. It is used to predict the genotype of offsprings if the genotype of parents is known. So this Punnett square, this chart, it's known as Punnett square, and this Punnett square, it is used to predict the genotype of offsprings if the genotype of the parents is known. Okay, now we have two parents here, capital G, and a small one plant with green color pods with the genotype capital G, and another plant here with yellow color pods and with the genotype small g. So we use this Punnett square to predict the genotype of offsprings, and from this Punnett square we concluded that all the plants in F1 generation will have the genotype capital G and small g. That's what is the applicability of the Punnett square. Now here again, let's see this. In case of, here we had two gametes, capital G and capital G. Here we had small g and small g. Now all offsprings will have green color pods because capital G is responsible for the green color of the pod and it's dominant toward the recessive allele small g. So all the offsprings in F1 generation, they will have green color pods. And now if we self them, if we subject these F1 plants, F1 heterozygotes or F1 hybrids to selfing, here again we'll see. If you will rub this first. uh, here if you look at this, we have the F1 hybrid has the genotype capital G small g, and if I subject this plant to selfing, we will have two alleles. This is one parent and this another parent. Now here we'll have two gametes, one will be capital G and small g. Here again we'll have capital G and small g. Now we'll cross them with the apple pass. This Punnett chart will use this Punnett square. Now we'll have capital G small g on one side and capital G small g on one side. Now if we see we cross them, this will be capital G and capital G. This will be capital G and small g here, and then we'll have this capital G and small g here, and we'll have small g and small g here. Now here in this case we have three plants which have green color pods. This 1, 2 and 3. So three plants in the F2 generation have green color pods and one plant has yellow color pods. But if you look at this, the plant of the parents were both capital G and small g. They were both green color. They were having green color pods, but in the F1 we have one plant which has yellow color pods. So what was in these plants, what was in the parents that we have, what was in the green color uh, parents that we have one yellow color one plant with yellow color pods in the F2 generation? It was this allele small g and small g which was present in the parents. And this is what law of segregation says, that these two alleles which were together in the parents, they separated and then they joined randomly to form this. Had they been if they were not separated, if they would have not been separated, then we would never have yellow color then we'll never have yellow color pods in the F2 generation. But because they are subject to the law of segregation, because the two alleles capital G and small g, they separate from each other, that's why we have small g and small g, a plant with a genotype small g and small g in the F2 generation. Now again, if we look at the genotypic and the phenotypic ratio, we again look at the genotypic and the phenotypic ratio, we'll get the same results. Now if we here we have the character is color of the pod. Now if we look at the phenotypic ratio first, phenotypic ratio. Now phenotypic ratio means external appearance. We have one is green, this is again green, and this again. So green color we have 3 and yellow color we have 1. So based on this we have the phenotypic ratio of 3:1. Now if we look at the genotypic ratio, GG is only once. So here we have GG only once, so it will be 1. Now capital G small g is twice. Capital G was once. Now we have capital G small g is twice, so it will be 2, and small g is once, so the genotypic ratio will be 1:2:1, and the phenotypic ratio will be 3:1. So this is another example of monohybrid cross, and this explains the law of segregation very well. So finally we'll revise what law of segregation says. Law of segregation says, or law of segregation states, that the two alleles of a gene which are responsible for a particular trait, which are responsible for a particular character, separate during meiosis or separate during gamete formation. This is what law of segregation states. Now when we move to the next lecture, we will study the law of independent assortment. So stay connected. Thank you and have a nice day.